So it's a great pleasure for me to introduce the first speaker, Professor Tyler Jacks. Tyler is director of the Koch Institute at MIT. He has pioneered the use of gene targeting models to understand genes that cause cancer, in particular the P53 and the RB family of genes that have really dominated the field for so many years, actually decades. More recently, he's turned to gene editing, and I think he's taken this to a new level by exploiting mouse models in order to understand genes that drive the driver events of cancer. So, of course, he's received many, many accolades throughout his career, but to be very brief, he's a fellow of 3 academies in the US, including the National Academy of Sciences, and he currently chairs the National Cancer Advisory Board in the US. So welcome, Tyler.
Well, thanks very much, Jane, and thanks to the organizers for including me in this very special occasion at this very special institute. I have lots of friends who have been trained here at the WEHI or work here at the WEHI or both. So it's a real pleasure for me to participate, and I thank you for allowing me to be here. So my lab is interested in cancer, and we use mouse models to study the disease. And the reasons for that are somewhat illustrated on this slide. We know that cancers arise from normal cells through mutations in cellular genes, oncogenes and tumor suppressor genes, and we can use genome editing and genetic engineering technologies to introduce mutations in these genes in the mouse and study the effects in a tumor cell intrinsic fashion of the consequences of those mutations. But I think as importantly, we know that cancers arise in context. They interact with many other cells of the body, and these interactions are very important in dictating the behaviors of the tumors, how well they'll progress, how well they'll respond to therapy, what the ultimate outcome will be for the individual. And for these reasons, it's important to look at cancer development in context. And there are relatively few systems in which one can do that. And because of the genetic tools that exist in the mouse, we think that's the appropriate place to do it. Not the only place, certainly. This field goes back many, many years. The first genetically engineered mouse models of cancer were developed in the early to mid-1980s, following transgenic technologies pioneered by Palmater and Brinster. And an early example of the use of this technology derives from this very institute. As you've heard in previous presentations, and I'm sure you already know, MYC transgenic mice have been used to study the consequences of MYC and tumorigenesis in various settings, and this very important piece of work from Jerry Adams and Suzanne Corey and Alan Harris, and importantly the graduate student on the project, my friend Lynn Corcoran, was one of the first and very influential papers in this field. Many developments have occurred over the years in our ability to make other types of mutations and use other technologies to engineer the cancer genome, and we've exploited these over the years to construct a whole series of mouse models. I'm going to focus today on models of lung cancer, in which we've used 2 very important cancer genes that are mutated at high frequency in the human disease, KRAS, the oncogene, and P53, the tumor suppressor gene. And we use Crelox technology to activate an oncogenic allele of KRAS in its endogenous locus and to inactivate the 2 wild-type copies of P53. And we do that using viruses that carry Cre recombinase, and these days we typically use lentiviruses. And that's important, by the way, because as you'll see in a couple of examples, we can bring other genes in on the back of the lentivirus, and in that way influence the genome of the cancer cells from its earliest stages of development. This shows you the histological progression of this form of lung cancer. It looks like adenocarcinoma of the lung in humans. It goes through stages from hyperplasias to adenomas to frank cancers, adenocarcinomas, and in about 30 to 50% of these animals, we observe distant metastases. And we've been trying to use these models now over the last several years to understand the various stages of tumorigenesis, and also, as I've mentioned, the important interactions that take place between the developing tumor and the cells of the host. And the first of 2 stories that I'll tell you today focuses on one of those types of interactions. And I chose to tell this story at this talk because of the great strengths that exist here at the WEHI in immunology. And I'm telling you this in part to encourage a convergence of 2 great strengths here, actually, cancer genetics and cancer models, and immunology as well. Cancer immunology, as you know, is a very, very exciting and very important field, partly based on the fact that there are new therapies in the clinic which utilize inhibitors of the things that control the immune response in cancer. But there are many questions left unanswered, and I think the great strengths of the WEHI could be brought to bear to answer some of those questions and to advance that field. So I encourage you to at least think about that. Now, importantly, as we got started trying to explore the connections between the immune system and a developing tumor, it was important to ask, does the immune system even see these tumors as they develop in these mice? We and others actually hadn't looked very closely at the natural immune response to a developing tumor in a genetically engineered model of the disease. So Michael Dupage in my lab some years ago simply asked, as tumors developed, can we see an immune response by just looking at infiltration of T cells and B cells into the tumor? And the answer, I would say initially, to our surprise, was no. If you look at several weeks after tumor initiation in this model, where the virus introduced has CRE only, so RAS gets activated, P53 gets inactivated, you see very few T cells in the tumor, very few B cells in the environment of the tumor as well. So these tumors are not very immunogenic, as it turns out. I'll also tell you that if you inactivate the immune system by getting rid of the RAG genes, there's no effect on tumorigenesis. So again, that's an indication that in this setting, the immune system is not controlling tumor development. We think we understand that now because if we've sequenced the genomes of several of these tumors, or at least the exomes of several of these tumors, and in fact, they have very few de novo mutations. On average, these tumors have about 12 protein-altering mutations, and so given that very small number, you'd expect that very few, if any of them, would develop strong epitopes that the immune system would actually see. So in fact, these tumors are somewhat stealth to the immune system, and therefore it's not surprising that the immune system does not see them. So the question is, what can we do about that? Oh, I'm just realizing that that's horribly out of order. I apologize. Michael Dupage in my lab decided to augment the system by introducing a specific antigen into the developing tumors. And he used this approach of creating a bifunctional lentiviral vector that has CRE on the one hand and then has another gene, here luciferase, which has been fused to antigens that are known to be seen by the immune system. In fact, he had two different ones, synfecal and sergal, the OVA1 antigen and the P1 antigen. And in this way, we could program the tumors to express an antigen, and then we could ask in that context, do we see an immune response? This work was published a few years ago, so I'll just summarize the major findings. Importantly, in this context, the immune system does see the developing tumor. We could measure an antigen-specific immune response arising in these tumor-bearing animals, and we could see that actually at the level of infiltration. If we now just do lymphocyte staining inside the tumor, we could see pretty substantial immunoinfiltrates. And importantly, that was a productive response, by which I mean the antigen-expressing tumors developed less well than their non-antigen-expressing counterparts. The immune system was controlling tumor development. And we knew that was a specific effect of the immune system because now if you do this experiment in a RAG mutant background, that effect goes away. So the immune system can see a developing tumor in these genetically engineered models, but you have to provide an antigen, not surprisingly. The bad news is that that effect does not last. Although it's true that at the early time points, a couple of months after tumor initiation, you see infiltration and this effect. If you wait another couple of months, the effect goes away. Now one does not see an immunoinfiltration into the tumor. If anything, there are T cells in the boundaries or the margins of the tumor, and these tumors then continue to grow and develop equally well to their non-antigen expressing counterparts and kill the animals with much the same kinetics. So there's clearly some form of immunoevasion or immunosuppression going on here. Importantly, this is not classically immunoediting. The antigen remains expressed in the developing tumors, but despite that fact, the immune system gets shut off. There's some form of T cell exhaustion taking place, and we can demonstrate that based on the activity state of the T cells and their numbers, and also with respect to infiltration. So the question is, what is the basis of that effect? And now again, my out of order problem exists. There are several possible mechanisms to account for this form of immunosuppression. You've all heard about the various checkpoint factors like CTLA4 and PD-1 and PD-L1. Importantly, in these tumor-reactive T cells, we see the upregulation of these checkpoint proteins like PD-1. We've actually used anti-PD-1 antibodies, and we can't actually restore an immune response against those. So we've considered other factors that might be controlling the immune response, and as you know, there are many. There are many cellular factors in the tumor microenvironment that can also inhibit an active immune response. Myeloid-deprived suppressor cells, for example, certain forms of macrophages or dendritic cells can suppress an immune response. And T regulatory cells, likewise, can control an immune response in the periphery. And you've already heard from Daniel Gray this morning about these Treg cells controlling immune responses in the periphery. And they're known to be important in the cancer context as well. And we wondered, might they be important in controlling the immune response in this model system? Daniel told you about the functions of Tregs. They derive from FOXP3 positive cells, and they control immune responses in the periphery by regulating antigen presentation on the one hand and directly suppressing immune responses in lymphocytes as well. So they're a very potent cell type. The question is, are they important in controlling the immune response in this model system? And the first thing we wanted to know was, could we observe an increased number of T regulatory cells in these tumor-bearing animals? So Nick Joshi, who did this work, simply used FACS staining and immunostaining as well, and demonstrated that indeed in tumor-bearing animals, as shown here, there's a significant increase in the lung tissue surrounding the tumor of T regulatory cells as these tumors progress over time. And that's quantified here. So Tregs are there. So that's guilt by association, but are they really important in controlling the immune response in this setting? And fortunately for us, we were able to ask that question directly because Sasha Rudensky's lab had made a mouse in which they knocked in the diphtheria toxin receptor into the FOXP3 locus. And with this animal, one can deplete Tregs using the administration of diphtheria toxin. And so Nick crossed this FOXP3 DTR allele in with our KP cancer genes, along with tomato. This is a tomato reporter that allows us to see the tumors more readily. And in this context, using the virus that expresses the specific antigen, we could make tumors that are antigen expressing, allow them to get to the point where we observe immunosuppression. Tregs are still present here. And then we could add diphtheria toxin and do depletion very acutely. And this is actual data that shows that we can get very nice Treg depletion in this setting. And then at this time point, at about 12 days post-depletion, we can ask what is the effect on the tumors following the loss of Tregs. We have to look at 12 days because at 14 days, the animals are dead. And that's because, as you heard this morning, when you get rid of Tregs systemically, there's massive autoimmunity. So we have a relatively short window to assess the consequences of Treg depletion in this model. So what happens? Well, this is a before and after picture. This is the before. You can see here a tumor at 20 weeks. This is an antigen-expressing tumor. Here's the tumor, nice and cherry red. Here are the T cells, and they're present right at the margins of the tumor, as I mentioned. This is typically where we find T cells at this later stage of tumor development. They're near the tumor, but they're not in the tumor. I'll come back to that point in a minute. Tregs are still present here. They're actually right there as well. When we get rid of the Tregs using diphtheria toxin administration, this is what we see. The tumors are overrun with T cells. And those T cells are composed of both CD4 positive and CD8 positive T cells. They're not the only immune cells in the tumor as well. There are macrophages there. The T cells are active. They're killing the tumor, and the macrophages are presumably cleaning up the junk. And this is what it looks like histologically. This is a control tumor, nice and healthy tumor cell stained in brown. And this is following Treg depletion. Again, you can see substantial loss of tumor cells and effacement of lymphoid cells. So Tregs clearly are controlling the immune response in this setting, and we think, therefore, that there's some potential, if we can figure out how to do specific inhibition of Treg function in the context of a tumor, this might be a new therapeutic avenue for immunoregulation of cancer. That's a challenge, of course, because we can't get rid of Tregs everywhere because of the autoimmunity problem, but we're looking now at the Tregs in the tumor microenvironment. Do they have pathways that are active, gene signatures that are on, that might allow us to give some specificity to the inhibition of those cells? Before I switch gears, I wanted to just return to this subject of where this is taking place, and this is really for the immunologists in the group. As I mentioned, at this late time point, we do see T cells in the environment of the tumor, but they tend to be on the outskirts. In fact, they tend to be organized in these tertiary lymphoid structures. These antigen-expressing tumors almost always have one of these tertiary lymphoid structures associated with them, and those tertiary lymphoid structures are composed of B cell zones and T cell zones, and they've got other hallmarks of lymph nodes. And the Tregs, as I've mentioned, are there as well. We think these structures are probably an important site of antigen presentation on the one hand, in the early phases of an immune response against a tumor, and are also the site of immunosuppression at these later stages. As I mentioned, the Tregs are present there, and we can observe the effects directly within these TLSs following Treg depletion, looking, for example, at proliferation of the lymphoid cells within these TLS structures. Prior to Treg depletion, when we do BRDU staining of these sections, or of these animals, we can see the occasional BRDU positive cell in white. Following Treg depletion, these TLS structures light up like a Christmas tree. The T cells are being actively stimulated to proliferate and then presumably infiltrating the tumor and causing tumor destruction. So this is an example of how to use mouse models to study details of the immune response to a tumor. Many, many questions still to ask here in this specific story and related ones. And again, I encourage you to think about doing that here in your own labs. Let me switch gears now and focus my attention not on the interactions between tumor cells and the cells that surround them, but rather the specific changes that take place within the tumor cells themselves, the genetic changes in oncogenes and tumor suppressor genes. And this is now a growing and ever more complex area. As human cancer specimens are sequenced by exome and genome sequencing methods, we're realizing that they're highly complex. They have many, many mutations and other structural alterations in their genomes, quite different from what I told you about the mouse model that we've created. And in fact, in lung cancers, and all of these studies I'm showing you here are lung cancer examples, tumors tend to have hundreds of protein-altering mutations. And this is somewhat bewildering, I must say, for those of us in the field. It's quite a challenge when we think about how to deal with that level of complexity and how to study it in the mouse models. Here's a specific example from Matt Meyerson and the Cancer Genome Atlas Network looking at lung adenocarcinomas. And you can see a large number of genes that are mutated at reasonable frequency. KRAS and P53 at the top of this list. That's good for us. Those are the two genes that we modeled in our mouse model. But you can see there are many other genes that are mutated as well. And for those of us who do this sort of work and sort of bringing functional genomics to this genomics area, it's a daunting task. And the reason for that is, as you guys well appreciate, if you want to do functional validation of a gene using traditional methods, you would have to either acquire from a friend or make in your own lab a mouse with a mutation in that gene, and then cross it into your favorite cancer-prone strain, work that up into a large cohort of animals, induce tumors, and then assay the effects. This would take you years to do and untold thousands of dollars per gene, let alone the tens or hundreds of genes that are being identified from the Genome Project. And so this was a dark and depressing time for us. And in our despair, we looked to the heavens. And fortunately, the clouds parted, and delivered unto us was Crispr. And it was good. So Crispr has been introduced by other speakers. You're all familiar with it. It's an ancient system of bacterial immunity that turns out to be reliant on just two key factors, an enzyme that cuts the DNA called Cas9, and a guide RNA that can be designed to bring that Cas9 to any sequence in the genome that's next door to a simple 3-nucleotide PAM sequence. Thanks to the work of Jennifer Doudna and Emmanuel Charpentier, we now know that we can construct systems just based on these two components, port them into mammalian cells, and get them to work quite well. As you know, following the cutting by Cas9, repair pathways take over to repair the damage. If you don't apply any other material to the cell, it'll undergo non-homologous end joining to fix the break. That's an error-prone process, which will also often give rise to insertion and deletion mutations, loss of function mutations, and that's good for cancer modeling studies. If you provide a homologous recombination donor sequence, you can transfer mutations from that sequence into the genome at that site. And this technique has been used now over the last few years to do many, many very important studies in the cancer world to create new strains of mice, ES cell variants, cell-based experiments, lots of screening being done. It's a very exciting time using this technology in cancer research. We decided, we asked the question, could we use this technology to accelerate functional genomics in the whole animal? Could we use CRISPR technologies to mutate cancer genes directly in the cells of living mice? And would that allow us to accelerate the pace of functional genomics? And fortunately, the answer is yes. In the first study that we did, which we published last fall, and I'm just summarizing for you here, we took the simplest approach, and that was to take a plasmid from Feng Zhang's lab that had Cas9 and a guide RNA that we could program against a gene of interest, and then just inject that plasmid directly into the tail vein of mice and rely on hydrodynamic gene transfer to deliver that DNA into the livers of those mice, which happens at relatively high efficiency. And we were able to show in this study that if we used a guide against P10, we could develop P10 deficient cells that we could see by histology and by deep sequencing. If we combined plasmids that had a guide against P53 and P10 and delivered those into mice, we could develop cancer in these animals just 3 months later. And if we did deep sequencing or just sequencing of the DNA of these tumors, we could find biallelic mutations in P10 and P53. So single-step mutagenesis in a wild-type mouse giving rise to a tumor in relatively short order. We could also do gain-of-function mutations here in beta-catenin. So the guide is against beta-catenin, but we also added an oligonucleotide that brought in 4 base changes that converted beta-catenin into an oncogene. And in this case, at low efficiency, we could observe cells with nuclear beta-catenin, which we could confirm then had those 4 base changes. So you can do loss-of-function mutations, you could do gain-of-function mutations really quite easily. With the success in the liver, a tumor that we don't otherwise study, Francisco Sanchez Rivera in my lab and Thalis Papagiannakopoulos decided to ask, can we make CRISPR work in our lung cancer model? And for that purpose, they designed this vector, which we call PSEC. It's quite simple. It has CRE to do KRAS and P53, but it also has Cas9 and has a site to introduce a guide RNA against any gene of interest. And in this proof of principle study, we chose 3 different tumor suppressor genes, NKX2.1, APC, and P10. And the question was, if we introduce this vector into animals, will it accelerate tumor development as a consequence of the mutation in the tumor cells of these tumor suppressor genes? And the answer, also recently published, was yes in all 3 cases. So indeed, when you mutate NKX2.1 or APC or P10 along with KRAS and P53, you get more tumors per area than in the control animals, and as well, the tumors progress to a more advanced stage of pathology. So indeed, we can quite quickly confirm that these tumor suppressor genes have the expected effects by inactivating them in this directed fashion. This is what they look like, and I'll just show this quickly. We see what we expect when we get rid of NKX2.1. The tumors become quite mucinous. They actually look a little bit more intestinal when you get rid of this transcription factor. When you delete P10, you can see activation of the PI3 kinase signaling pathway in the tumors. And when you get rid of APC, one can see nuclear beta-catenin in these tumors as expected, as well as upregulation of SOX9, a change in differentiation state. In this study, which was completed about 7 months after it was conceived and about 5 months after it was started, we generated probably hundreds of tumor samples in what would have taken us, in a more conventional sense, probably 10 times as long, and obviously much more money along the way. So this has really transformed how we're prosecuting various genes of interest now in the lab. We're taking on long lists of cancer genes of interest and pursuing them using this strategy. We can look at the DNA of these tumors and assess the consequences of CRISPR-mediated mutation of the target genes. Here's an example in P10. Here's a tumor in which we see 2 predominant loss-of-function alleles. There's a 3rd minor allele as well. Probably most of the action is taking place in those very first few cell divisions, This is a very efficient process, but occasionally you'll see examples where the situation is more complex, like here, there's one predominant allele and then several minor alleles. This is a situation probably where very early on there was a mutation in one allele, but then several cell divisions happened where the mutations of the second alleles happened separately in the different descendants of those cells, giving rise to independent mutations. So none of the genes that I've just told you about actually appeared on this slide here. None of those tumor suppressor genes are actually mutated at high frequency in human lung cancer. The closest would be the PI3 kinase activation, so the analog of PTEN deletion, which does happen at some frequency. So we've wanted to now turn our attention to some of the other genes that are mutated at reasonable frequency in the human disease. And the one story I'll tell you about briefly involves this gene here called KEAP1. KEAP1 is the third most frequently mutated gene in human lung adenocarcinoma. About 17% or so of human adenocarcinomas carry mutations in KEAP1, and these are loss of function mutations. KEAP1 is a tumor suppressor gene in this disease. You may or may not know about KEAP1. It's an important negative regulator of an antioxidant program that cells have. KEAP1 negatively regulates a transcription factor called NRF2, and NRF2 is required for cells to deal with increased reactive oxygen species. And as tumors develop, partly based on their increased metabolic rates, partly based on just direct oncogene action, they tend to build up levels of ROS species. And the cells respond to that by increasing their antioxidant capabilities, in part by increasing the levels of NRF2, this transcription factor. The pathway is shown here in outline form. Normally, KEAP1 binds to NRF2 in the cytoplasm, leading to its degradation. When KEAP1 itself gets oxidized through several cysteine residues, it releases NRF2, allowing NRF2 to go into the nucleus and transcribe a variety of genes, enzymes involved in cellular detoxification. So that's the pathway, and the pathway, as I said, is frequently mutated in human cancers, in particular lung cancer. In the case of lung adenocarcinoma, KEAP1 at about 17%. NRF2 mutations also occur in this disease, and the NRF2 mutations are gain of function. They're mutations that affect the domains that normally allow NRF2 to be bound by KEAP1. In lung squamous cell cancers, you find an even higher frequency of KEAP1 and NRF2 mutations. About a third of these tumors carry mutations in these two genes, again involving KEAP1 loss of function and NRF2 gain of function. David Tuvison has actually studied NRF2 specifically in lung cancer as well as pancreas cancer. He used a germline mutant of NRF2 to ask whether loss of this important detoxifying gene would affect tumor development. The expectation would be that it would inhibit tumor development. The model is you need NRF2 to deal with the increased ROS. Loss of NRF2 should block that effect, and that's exactly what Dave saw. In NRF2 mutant animals, both in the lung and in the pancreas, they developed fewer tumors. So Francisco and Thales wanted to ask the question using our PSEC system, what would we see? And to specifically ask what would happen if you delete KEAP1. And as a control, they also targeted NRF2 to see whether they could regenerate or reproduce the Tuvison effect that I just told you about. And with that system, they could do a lot of downstream experiments, obviously looking at tumor development as a first case, but also looking at some of the biochemical effects that you might expect to see in these tumors. So what happens? Well, in the case of KEAP1 loss, indeed we see an increase in tumor development. KEAP1 is thought to be a tumor suppressor gene. It had never been tested and shown to be a tumor suppressor gene, but it looks like one here. In the animals in which we've targeted KEAP1 using the Crispr system, we see increased tumor volumes as measured by micro-CT imaging shown here and quantified here, and also increased tumor grade as shown here. So KEAP1 is a bona fide tumor suppressor, a very potent one actually. Very few genes work quite as well as KEAP1 in this setting. We can observe mutations in these tumors, again, somewhat typical biallelic mutations. Two predominant alleles, they're both loss of function alleles, an insertion and a deletion. Sometimes the picture is a little different. Here's a tumor with only a single mutant allele evident. This is probably a case where one allele got mutated and the second allele was simply lost by a chromosomal event. Now, if things are according to plan, then when you mutate KEAP1, you should have hyperactivation of NRF2 and increased expression of NRF2 target genes. And that's exactly what we see. One NRF2 target gene is called NQ01. It's a quinone reductase. And indeed, in the majority of the KEAP1 deficient tumors, we see upregulation of this target. So this looks exactly as we would expect. And we're doing lots of experiments now to explore and investigate the biochemical changes that are taking place within these tumors, and also hoping to find certain vulnerabilities of these tumors that we can exploit therapeutically. Now, in closing, let me tell you about this situation, which took us by surprise. I mentioned that we all targeted KEAP1, where we expected to see increased tumor burden, but we also targeted NRF2, where we expected to see a reduction in tumor burden, because the model was that you needed NRF2 to deal with this increased ROS. But in fact, as you can see here, the NRF2 targeted tumors also showed increased tumor burden and increased tumor grade. And that was a surprise to us until we remembered that NRF2 is also targeted in cancer. It's also mutated in cancer. I've mentioned that to you. NRF2 undergoes mutation in human cancers, but these aren't loss of function mutations. They're gain of function mutations. They're point mutations in the residues that NRF2 normally uses to bind to KEAP1. And so this raised the possibility that these tumors were actually selecting for mutations that looked like this. And it turns out that just by coincidence, the guide RNA that we were using to target NRF2 targeted that very region. And it turns out that the tumors that were arising in these animals had carried deletion mutations. Here you can see the DNA of one such. This is a 342 base pair deletion. It's an in-frame deletion that would be expected to produce a stable protein, which it does in those tumors. And that protein is functional and turns on NQ01. So this is an example of forward genetics, actually. The tumor is selecting for the kinds of mutations it needs to progress. Here, ones that liberate NRF2 to basically phenocopy loss of KEAP1. If that idea is correct, if we use a different guide that targets a different region of the protein where that can't happen, perhaps we will see the expected effects. And so Thalys and Francisco targeted a different region here in exon 2, and in this case we observed a decrease in tumor development as expected. So I'm going to close there and just summarize this approach, which we're very excited about. It's very valuable for studying genes in the lung, for using the methods that I've described for you. We also know that it can be used in other sites. The same PSEC vector has been used by Julian Sage to target genes in a pancreas cancer model. Omer Yilmaz in our institute has worked with us to target colon cancer genes using the same system. And there are likely to be many other places where the same system can be used to accelerate functional genomics in cancer. In the future, we imagine doing even more complex types of editing with multiple guides targeting multiple genes of interest, using vectors that carry Cas9, or indeed mice that carry Cas9 in the germline, which now exists thanks to Feng Zhang. We've made one too. And also using the modified Cas9s, the dCas9s, that will allow activation of genes of interest or silencing of genes of interest by different mechanisms, as well as now perfecting the methods for homologous recombination and the activation of specific mutations using these methods. This is not quite so straightforward because you need another piece of DNA to make this work, but we think with different viral vectors or nanotechnology-based approaches, we'll be able to make this work as well. So I'm going to stop there and thank the people who've done this work. The immunology was pioneered initially by Michael Dupage, and all the Treg work that I told you about was carried out by Nick Joshi. Wen Zhu was the postdoc who got Crispr to work for us initially in collaboration with Feng Zhang and Phil Sharp and Dan Anderson. And that work was carried on further into the lung by Francisco Sanchez-Rivera and Thalys Papagiannakopoulos. Thanks very much.
Thank you for a very exciting talk. We have time for a few questions.
Jane, why don't you do it?
What pitfalls do you need to be aware when working with Crispr?
What pitfalls? So I could answer that question on a number of different levels. Experimental pitfalls would include off-target targeting. And so that's an important question. We've actually looked at that. We've in the case of several of those guide RNAs, we've looked at the mutation frequency of predicted off-target sites and actually have seen very little, very few examples of off-target cutting and off-target mutagenesis. So the system looks relatively clean. Having said that, there's another layer of concern you might have, and I think it's important to point this out. We use lentiviruses that carry Cas9, the enzyme, and a guide RNA that's enough to bring Cas9 to the target and cut it. If that same guide RNA works against the mouse and human, there's a safety issue, something that we now worry quite a lot about, having discovered that one of our very potent guides actually does target the human gene itself. So I think it's important to pay very close attention to how these vectors are being handled and make sure they're being handled in a safe way.
This is a really interesting talk. In the data you showed about the tertiary lymphoid structures, do you think it's possible that the same Treg population is responsible for the initial activation and then the subsequent suppression of that T cell response to the growing tumor?
I don't know the answer to that question, in part because we've not studied the development of those TLSs in great detail yet, but we're doing that right now. I think it'll be important to sort of map out their natural history as the tumors are developing and the immune response is both productive and then inhibited. So I just don't know. I don't know exactly when in the development of those structures the Tregs arise, but it is possible, of course, if they're there early, that they could be positively affecting the immune response.
So with the second guide RNA against NERV2, where you did see reduced tumor burden, in the small tumors that you found, was there potent selection against that guide RNA?
We're just looking at those tumors now, so I don't know. But yes, of course, it's possible that those will be examples where there's been escape through one of a number of mechanisms. And again, you could create in-frame mutations that allow still function there. So we don't yet know. But yeah, so I can't answer that question.
Tyler, so the current thinking, I guess, is that there are 3 to 5 genetic drivers for lung cancer. Well, what about a role for the other mutations? Do you think that they're influencing, say, a 10%, 20% frequency along the lines of Crispr Now's talk this morning? Or
So you're asking, with respect to human cancers,
yes,
how many genes that we find mutated are actually contributing to
3 to 5
Yeah.
genetic drivers per tumor?
Yeah, I think that's probably about right. If you look at the sort of, in a given tumor, how many genes are mutated that you expect to be. I think it's probably, in the case of lung cancer, a little bit higher than that. But drivers and influencers are two different things. So there might be genes that are being selected for that don't profoundly affect the tumor phenotype, but nevertheless influence the tumor phenotype. For example, influence the immune response to the tumor, that are nevertheless contributing.
Thank you once again. We'd better move on.